The life cycle of the root knot nematodes. The mobile, second-stage juvenile, is attracted by the growing root tips, usually near the apical meristem, zone of cell elongation, and near the points of emergence of lateral roots. It penetrates behind the root tip, in the zone of elongation. Once it establishes a feeding site in the primary phloem, or adjacent undifferentiated parenchyma cells, it begins to feed, and increases in diameter, where it loses its ability to move, except for the extreme anterior end of its body. In response to the feeding of the second-stage juvenile, the host tissue undergoes pronounced morphological and physiological changes. Some proxylem cells develop into permanent giant cells for the nematode, that are formed by repeated mitosis without cytokinesis. These hypertrophied, multinucleate, giant cells act as a metabolic sink, producing large amounts of proteins and other molecules that are for the benefit of the juvenile rather than that of the plant. It rapidly molts to the non-feeding third-stage juvenile. The third-stage juvenile rapidly molts again to form the fourth-stage juvenile, where the male morphs back to the vermiform shape. The male fourth-stage juvenile molts one last time, and becomes a mature male, which leaves the root gall to look for a nearby female, with which to mate. Shortly after the fourth and last molt, the adult female resumes feeding, and swells up in diameter, sometimes bursting through the endodermis, to expose her posterior end to the soil. She begins to secrete a gelatinous matrix, in which she will deposit her eggs. Eggs within the egg mass undergo embryogenesis, from that of a single cell, to a fully formed first-stage juvenile. This stage undergoes its first molt inside the egg, and hatches as an infective, second-stage, juvenile. These second-stage juveniles migrate through the soil, to invade waiting root tips, where their life cycle starts all over again.